Hey guys, and welcome to this week's video, and today it's going to be all about interphase, which is part of the cell cycle. Now this is going to be part of a series that I'm making about mainly mitosis. However, interphase is, I like to say, a prequel to mitosis, allows mitosis to occur, but it's not actually part of mitosis, so we're just going to include it under the large umbrella of the cell cycle. And this video is going to be focused on the IB diploma and curriculum. However, again, like I've said countless times before, everybody's welcome to watch, and the information shared is still valid for general knowledge. So the first thing to know about interphase is that it is the phase in which most cells actually spend their time. So as you can see here, 5 to 10% is actually the M phase, which is mitosis, which is when the cell actually divides. And then just a little bit, not even significant enough to have a percentage, is cytokinesis, when the cell actually splits physically. But most of the cycle is interphase. So most of the time, the cells that you see under a microscope or the cells in your body currently are in interphase. And the general function or the general components of interphase include that the cell performs its normal functions like producing energy, secreting some surfactant or whatever its function is for the body or tissue where it is. However, in interphase, the cell also will start to prepare for cell divisions. It grows, the cell size increases, and it duplicates its cellular components such as organelles, for example, more mitochondria. And in interphase, what you have to know about the DNA or the chromosomes is that they are decondensed. And decondensed, I mean they're really, really messy. No organization. You're used to seeing chromosomes in nice little pairs or homologous chromosomes, so to say. However, in reality, most of the time our chromosomes are coiled like I have drawn here, and they're actually not called chromosomes then, they're called chromatins. So this is a term that you should get familiar with. And lastly, what occurs in interphase is also the DNA replication. So in preparation for mitosis, in order for the two sister chromatids to be able to be pulled away from each other, as we will see in mitosis, we need to actually create these two sister chromatids, which we do through DNA replication. But more on that later. The result of DNA replication, like I've written here, is that two sister chromatids are created that are attached at the centromere. So this is the first sister chromatid, this is the second one, and here there's a centromere. Now, this is a very general description of interphase. And when we go into specifics, we can actually further divide interphase into 3 more stages: G1, S, and G2. And just to help a little bit, I'm going to draw arrows to see where you're going, but again, the numbers help. G1 comes before G2, and in between there's an S. So G actually stands for growth, so that might really help you remember what these phases are all about. So in the G1 phase, at the beginning, we have a really small cell. As you can see in the cell cycle, it just left cytokinesis, meaning that it's a really small daughter cell of a larger parent cell that divided into 2. So during G1, the cell will form membranes, produce organelles, it will produce proteins and molecules that it needs to sustain itself and to perform its function, and it will also increase in size, as you can see in this diagram here. So in this phase, the cell is just performing its function and getting ready to perform its function for the body. Now comes the S phase. The S phase is synthesis, and it is composed of DNA replication. Here we have in the beginning just one sister chromatid per chromosome. However, then we're going to replicate them and create 2 sister chromatids per chromosome. Now, be careful, we still have the same number of chromosomes because this and this are both one chromosome despite having 2 chromatids versus 1 chromatid, and that's because these are genetically identical, so you still have the same amount of genetic information. So both of these are diploid cells, they both have the same chromosome number, it's just that here you only have 1 copy, but here you have 2 as seen through the sister chromatids. And also one thing to note, that this still happens in the disorganized form of DNA, so it's still chromatin. I've just drawn it like this so you can see the actual replication. Now, the last part is the G2 phase. The G2 phase is the preparation of the cell for mitosis itself. So in G1, we prepared to actually function and sustain the cell, but in G2, the cell is preparing to divide. So we can see this through the division of organelles so that it can have sufficient amount of organelles in both daughter cells that it will split into. The cell increases in size, again, increase in cytoplasm volume, and overall prepares for mitosis, for example, centrioles over here and whatnot. And so these are the general phases of mitosis: G1, S, G2. In the end, you have a cell that's ready to divide, ready to go into mitosis. Now, let's talk about what facilitates the transitions between these phases. And of course, it's signaling, cell signaling. And this is mainly done by cyclins, which are proteins that signal to the cell to move from G1 to S to G2, and their concentration is low at the beginning but increases over time. And there are different types of cyclins that are responsible for different type of transitions, but overall they're under the umbrella of cyclins. And there are also CDKs, or cyclin-dependent kinases, which are enzymes. Kinase, A's should tell you that it's an enzyme, and cyclin-dependent already hints at its function, or hints at the way that it functions, and that is that it is activated by cyclins, right here, through a physical bond, which then results in a signal for the cell to move to the next stage. So from G1 to S, or to S to G2. And now lastly, I wanted to touch upon a G0 phase. Now this is quite interesting because this is kind of a special case of interphase, except it does not prepare the cell for mitosis. Only some cells enter G0 phase, for example, neurons, erythrocytes, and cardiac muscle. And if you already know something about these cells, you can maybe guess where I'm going with this, is that these cells never move past G1. So their entire function in life is just to execute its own role. It will never divide, and it will never enter mitosis. So we can say it exits the cell cycle, so to say. And for example, neurons, they don't divide, erythrocytes, they don't divide, and cardiac muscle cells, they all don't divide. So they never have DNA replication, they never go through the G2 phase, and they never enter mitosis. So I hope you enjoyed. I hope you learned something new about interphase, because this is quite a significant portion, if not the most significant portion time-wise, of the cell cycle. So knowing about it, a bit more detail, I hope was fun. And see you next week, and see you for the rest of the cell cycle series.